Hello class, it's Mr. Thomas. Hope you're doing well. Today we are going to continue our basic genetics notes and follow that up with some Punnett square notes. So on your next new page, go ahead and write Punnett square notes. You also need to update your table of contents and follow along with me as I walk you through Punnett squares. Additionally, you probably want to pause the video and get a purple colored pencil and a green colored pencil. So just to follow up what we learned about from our first basic genetics notes, Gregor Mendel, go ahead and write this down, Gregor Mendel is the father of genetics. That's right, he's the father of genetics. He's the guy that did all these famous experiments involving pea plants. Okay. And I'm just going to draw his experiment out for you so you have some understanding of what he did. So I'm just going to draw a flower here. He took two flowers, and remember, and again, if you'd like, again, you don't have to do this, but if you'd like, use some colored pencils to draw this out, or color this out, I should say. And he used purple flowers. Okay, so we have purple flowers. I guess that's kind of a pink, but that's purple. And then he crossed those purple flowers. Okay, I'm gonna make this a green stem and a purple flower. And I'm gonna just have you again cross that with a white. These were two pure parents. This time we don't have to color the plant flower, but I'm going to color the stem. And again, use colored pencils for this. Typically in class I'd use colored pencils, but I'm just trying this new note-taking method. It's not the greatest because my handwriting's kind of suffering here, but that's okay. So when he crossed a purple flower with a white flower, he found that the next generation got, and in class I would ask this question, I'd ask you, what did he get? And if you remember from what we saw there in the first video, he got 100% purple in the next generation. Okay, so go ahead and draw that out. The next generation, he got 100% purple flower color. And I like to draw it out so we remember it well. Purple crossed with the white yields 100% purple. Now let's talk about some of his vocabulary. Okay, this is the P generation. Okay, and P stands for pure parents. Pure parents. So we have a pure purple crossed with a pure white parent, and that generation yields what we call F1 generation. Okay, and this was 100% purple. So to show that, we used what is called a Punnett square. And a Punnett square is used to predict outcomes It's a device used to predict outcomes. After the fact, we can see what happened, but the pure parents have what we call genotypes. So a pure purple would be big P, and a pure white would be little p, because we used the dominant trait. We figured out what the dominant trait was from the F1 generation. The F1 generation was all 100% purple, so purple's got to be dominant. So to set up a Punnett square for the P generation, okay, this is the P, or the parental generation, the parental cross. We take, you're going to draw your box. I like to draw my box fairly big. Maybe draw, again, sorry, my lines are a little shaky using this tablet. It's fairly sensitive. It's a lot better than drawing with a mouse. The stylus gives me a little bit more comfort, but it's not how I typically write on paper. So we take a parent. So big P is our purple parent. And then we have a recessive parent, little p. I'm trying to exaggerate the size of my p's so that you know that the pure parent, the purple flower, is dominant. So I use big P, and so I take the top box and the side, the top line. My first plant will be big P, little p. Okay. Then I go to the next one, next box, the second box over, big P, still top line, lowercase p. And same for the bottom two squares. It's basically like reading a graph. And we get, notice in all four boxes we have the same genotype, okay? And let's go ahead and talk about this right now. These two letters, and again, it doesn't matter if you put the purple on the top or the white on the top. Those two letters represent a genotype. Okay. Each individual letter represents an allele. Learning to write better with this stylus here. But anytime I do a Punnett square in class, and to get full credit for my class, you need to do this. You need to decode your genotypes and your phenotypes. So off to the side, I will write genotype. And you can see here, we have four of the same, actually, genotypes. They're all big P, little p. So 4 out of 4 equals 100% big P, little p. Then off to the side, also write phenotype. And we have 4 out of 4 phenotypes. Because big P, little p is purple, 100% of our flowers are purple. So we'd write 100%. Okay, let's keep on going. Typically when I get this far on my page, if it was actual notebook paper, I'd be out of room. Okay, so, but that's okay. If you need to go to your next page, that's fine. If you want to hang with me on this page, just know we're going to do another experiment here. So we're now going to take these purple right here and cross them together. So I'm going to cross the purple I have up here, and I take those 100% purple. You know the genotype now, big P, little p. Two different alleles. and we're going to make the F2 generation. Okay, the F, or what's called an F1 cross. So our F1 cross is going to be 2 hybrid purple. Remember, hybrid is going to indicate that we have, and again, let me draw this out so you can see it. Draw a flower again. And this time, when I cross 2 purples together, and again, I said that correctly, it's not going to be 2 whites. I'm going to color these in right now for you. Cross a purple with another purple. Use colored pencils to show this. When Mendel took 2 impure purples, or what we call now as hybrid purples, and crossed them together, he got some interesting results. Okay, on the first cross, a lot of people during Mendel's time thought if you crossed a purple with a white, you'd get a blend, a pink. Okay, but we know that is not true. Now, after his experiments in the F1 generation, he got 100% purple. The interesting thing was not that, because that showed something entirely new. The interesting thing was that the F2 cross, or the F2 generation from the F1 cross, when he crossed a purple with a purple, he got purple again. But the strangest thing was that it wasn't just purple. He also got white flowers. Okay, and I typically ask this in class, you know, cross a purple with a purple, what do you get in the F1 cross for what's called the F2 generation? The F2 generation. And he got purple and white. But the crazy thing, it wasn't just that. He also got unique numbers, very specific numbers. Every time he did this, he got 75% purple and 25% white. Okay. Another way to say that is 3 to 1 purple to white. Okay. Because remember, we have big P, little p crossed with a big P, little p. And when you cross those together, in a Punnett square, which we're about to do right now, so leave space to do a Punnett square, and we'll kind of prove this. Okay, so again, with Punnett squares, what we do, we make a big enough box, 4 square box, show our letters. As we go on in class, these Punnett square boxes will get bigger and bigger, so it's best to learn it the right way. Every time we do a Punnett square set of notes, we'll add a new rule. But the basics work like this. You put an allele on over one box, the other allele over the other box. So in this case, I've got a big P, little p for my first parent. And I'm going to take the other parent. I'm going to put a big P, little p for that. Okay? And I'm going to cross them together. I get a big P in my first box, a big P, little p in my second box, a big P, little p in my third box. In my lower box, I'm taking just this allele and this allele, and I get a little p, little p. Okay? So again, let's decode this. Always write genotype and phenotype to decode. Alrighty, so we have 3 different genotypes. We've got a big P, 2 big P, little p's, and a little p, little p. So let's write that down. Big P, I always write every genotype down. That's 1 out of 4 boxes. So that is 25% of our genotype. It's going to be big P, big P. Our next box is going to be big P, little p. We have 2 of those squares in our 4, and 2 out of 4 is 50%. And then finally, we've got little p, and that is our recessive genotype at 1 out of 4, 25%. Okay, so we write that down, we decode it. Notice when we take these 2 genotypes, they're both purple. This is our pure purple, and this is our hybrid purple. Okay, the reason big P, little p is also purple is because it has at least 1 dominant allele. So we call this 75% purple, because obviously if you add 1/4 plus 2/4 or 50%, or if you just want to add 25% plus 50%, it comes out to 75%. Okay, 3/4 of our plants have purple flowers in a F1 cross, demonstrated by the F2 generation, and our remaining quarter is 25% white that exhibit both recessive alleles. Okay, and again, these words don't flow off the tongue easily until you use them. So I have an assignment coming up here where you'll actually read these terms out loud: genotype, phenotype, recessive, homozygous, heterozygous. So just so you know, big P and little p are both homozygous. Meaning they have the same alleles. One's homozygous dominant, big P, and one's homozygous recessive, little p, but they're both homozygous. And then in our F1 cross, okay, right up here, we had a big P, little p crossed with a big P, little p, and that is what we call heterozygous. Just so you can get your terms down. That is synonymous with a hybrid, okay, or impure. Some people call that impure. Heterozygous, hybrid, and impure, if you see those terms written, they're referring to the genotype where you have 2 different alleles. In this case, big P, little p. Okay, but this was Mendel's basic experiments involving pea plants. Again, just to review, you have the P generation, a pure purple crossed with a pure white, and he got 100% purple every time in the F1 generation. Then he took those 100% purple flowers and he crossed them amongst themselves with reciprocal crosses. Remember, if you watched the other or earlier notes, you'll know what that involved, cutting off the stamens and so forth. And he crossed those purple together, and he ended up with some purple and some white, but exactly 75% of those purple and 25% white. And we show that with a Punnett square, okay, to kind of show that outcome with that Punnett square. Going forward, we will use Punnett squares to make predictions, okay, of phenotypes and or of genotypes. For now, that's our Punnett square notes. Again, copy these into your notebook. We will be going into them in more detail as we move on. Thanks for listening, and this is Mr. Thomas signing off for now.